Welcome to Inherited Change. At AS level, your focus was on mitosis and genetic stability, the mechanisms that build identical clones. Now we shift entirely to genetic variation. This unit is the absolute cornerstone of A2 biology. You will bridge the microscopic world of DNA mutations with the macroscopic world of populations and evolution. It is a story of how traits are generated, shuffled, and passed on. Before we can predict how traits are inherited, we must understand how gametes are formed. We will track chromosomes through the precise stages of meiosis I and II. You will learn exactly why a reduction division is essential for sexual reproduction and how a single diploid parent cell divides to produce 4 genetically unique haploid cells. This division is the engine of genetic diversity. We will explore the exact sources of genetic variation, starting with crossing over. You will see how homologous chromosomes physically swap alleles at points called chiasmata. Combined with the random shuffling of independent assortment, you will calculate the immense number of possible chromosome combinations, explaining exactly why you are not an identical clone of your siblings. Next, we translate those shuffled alleles into physical traits. You will master the strict vocabulary of inheritance: genes, alleles, homozygous, and heterozygous. From there, you will construct flawless genetic diagrams. We will start with single-gene monohybrid inheritance before scaling up to dihybrid inheritance, where you will track 2 different genes simultaneously and learn to instantly spot golden exam ratios, like 9:3 to 3:1. But biology is rarely as simple as one dominant allele completely masking a recessive one. We will push beyond Mendel to explore codominance, where both alleles demand to be expressed equally in the phenotype. You will also tackle multiple alleles, using the human ABO blood group system to prove you can deduce parental genotypes from offspring phenotypes in complex dominance hierarchies. Why do genetic conditions like hemophilia and red-green colorblindness affect males far more frequently than females? You will uncover the rules of sex linkage, tracking genes that reside exclusively on the sex chromosomes. You will then apply this logic to real autosomal and sex-linked genetic disorders, learning how to decode complex family pedigrees and navigate the exact probability traps examiners use in Paper 4. Finally, we examine what happens when the genetic script breaks. You will distinguish between microscopic gene mutations, like substitutions and frame shifts that alter a single polypeptide chain, and macroscopic chromosome mutations. By understanding processes like non-disjunction, which leads to conditions such as Down syndrome, you will be able to explain both the causes of genetic disorders and the raw material they provide for evolution. By the end of this unit, you will have a complete, mathematically rigorous toolkit for genetics. You will be able to track DNA from meiosis to fertilization, predict complex inheritance patterns, and understand the molecular origins of genetic disease. Let's get started.